All right, so now we got to talk about protein structure. Protein structure. We typically break protein structure up into 4 levels. And we start with kind of the most basic elements of structure and work our way to more and more complex. And so the first level of structure we'll talk about is your primary structure. And we'll go to secondary, tertiary, quaternary. So that's with 4 levels. Notice we can also write primary structure like this. Same notation you might have remembered from primary, secondary, tertiary carbons in like organic chemistry. So but same diff, primary structure. So what is the primary structure ultimately? Yeah, just simply the chain of amino acids, your amino acid sequence. That's all it is. So, and we define the direction here. We look at the free amino terminus, and read it through that to the carboxy terminus. Notice you got to be careful, right? In this country we read left to right. In some countries they read right to left. So, and you got to be careful which way to read and stuff like that. Same thing with DNA. What is the normal way we read DNA? Oftentimes we define. So 5' to 3'. So it's all about direction here. Same thing here. We got to read it N terminus to C terminus. Notice that doesn't mean I can't draw it. You notice we read in this country left to right. And so normally when I write this, I put the N terminus on the left and the C terminus on the right. But nothing says that we can't just represent that and flip the whole thing over. And you're supposed to realize, oh, Chad put the N terminus way over here and the C terminus is way over here. I got to read it the other way. So be careful. We would always read it N to C, whether that means left to right, right to left, top to bottom, bottom to top. I don't know. It depends on how it's drawn, but it always is N to C. Cool. That is all it is. It's just amino acid sequence. So for a typical, peptide or protein, this can be, from several dozen to even hundreds or even over 1,000 amino acids in a row. So big, huge range in the size of proteins. So in this case, but important thing here. So these proteins, this is a big, long chain, So, and that big, long chain gets made where? It's not this class, but ribosome, right? So a big long chain gets all put together at the ribosome, and we think of it, as a big long chain, but truth be told, that big long chain doesn't stay a big long chain for long or even ever. It might actually start folding before it's even completely synthesized, but that thing's gonna fold up into some big 3-dimensional shape. So, and it turns out big key thing here is that all the information for how that thing is going to fold is contained within this amino acid sequence. We can actually take a protein and we can stretch it out until it's a linear chain and let go and fold right back up. Now sometimes it misfolds and stuff on occasion. Sometimes in the cell, proteins get help in folding and stuff like that, but all the information required for that thing to fold properly is in the amino acid sequence. It's not like here's a protein and now I need some other things in the cell to come up and fold it correctly and push that into place. It doesn't want to go there. Make it fit. It doesn't work that way. All the interactions, all the electrostatic interactions, the hydrogen bonding, the dipole-dipole forces, the hydrophobic interactions, all those things are all a function of this amino acid sequence, of the primary structure. All the information needed for folding is right here. Cool. So let's talk about our secondary structure. So your secondary structure. So some key things to remember about secondary structure. So it's beyond the amino acid sequence, it's the very first level of real folding that we're going to look at. So we got this amino acid sequence. Everything required for folding is there, but now we'll start talking about the most basic folding elements. So, and in this case, things you should know about secondary structure is secondary structure does not involve the R groups. It is only involved with the backbone of the amino acid chain. The R groups that are variable are not involved here. Cool, and the two most common are, what are the two most common secondary structure elements? Cool, alpha helix and the beta sheet, sometimes called the beta pleated sheet, same diff. Cool, those are those two most common secondary structural elements. They're not the only ones, but in many a biochem class and textbook, it's the only two that are ever discussed really in any detail. So there are others, but these are the only two we're going to talk about as well. So your alpha helix and your beta sheet, both of these structures are stabilized by hydrogen bonding of the backbone. So where on the backbone of an amino acid chain can hydrogen bonding take place? So if we look at a typical amino acid, We'll leave that out. So if we look here, hydrogen bonding can take place in a couple of different places. One, the oxygens have lone pairs. And having lone pairs, they can function as what? Donors or acceptors? They can be hydrogen bond acceptors. And so the oxygens can act as acceptors. Now if we look at the nitrogens that are tied up in peptide bonds, so those nitrogens, what could they act as? Donors or acceptors, technically. The nitrogen does have a lone pair, but in the secondary elements, we're only going to ever see them acting as donors. It's their hydrogen that is actually going to be involved. Cool. And so that's the nature of the hydrogen bonding. It's the backbone. Notice, are the R groups here part of this at all? Not this part. The R groups will be involved later, but for secondary structure, we're only talking about the backbone, not the R groups. So in this case, your alpha helix. So your alpha helix, so you take your long piece of DNA, your long piece of a peptide, and it starts spiraling around itself. And so on and so forth. And what stabilizes this is you might have, say, the oxygen at one part of the chain, and further on down part of the chain, you might have the nitrogen, the NH, on the backbone, and those guys interacting. That hydrogen bonding interaction is what stabilizes this. So if you notice, the axis of the coil, which I've drawn vertically, and the hydrogen bonding that's going on are parallel to each other. So we say that's important to remember. That's just a fact that makes an easy multiple choice test question. So in alpha helix, the hydrogen bonding is parallel to the axis of the helix. Cool. So a couple other random facts here. If you notice, they're in parentheses on your handout there. So your alpha helix, where are we at there? 3.6 residues per turn and 5.4 angstroms per turn. So from this point to the next analogous point in the coil as it goes up, so that's 5.4 angstroms, and it's 3.6 amino acids working your way around to get there. And this thing's just going to keep coiling up. And they're variable in length. They can be very short. They can actually be very long. So cool. But super common structural element to find in a boatload of proteins. A lot of proteins have these. In fact, I would say very few proteins out there. But you're going to find more that have these than that don't. So very common structural element. So then you got the beta-pleated sheet, or the beta sheet. So the beta sheet works a different way. So you end up getting long periods or long ranges of linear peptide. So, and then this peptide might come back and fold on itself and run the opposite direction right next to this. And what you'd find is that you'd have oxygens from the chain coming back, so in key places, and you'd have the NH in key places coming back. And so you'd have some hydrogen bonding interactions going on, say, here and here. So, and actually this guy would be over here. I can't draw this at all right now. This is terrible. And so on and so forth of the continuing chain as it loops back around, let's say. So, and if you look, that's what's actually drawn in a little text box on your handout there on the right-hand side. That's a beta-pleated sheet. Now, if you look, if this thing actually running N to C was going this way, and then just turned over on itself and came back, then these two strands, would they be parallel or anti-parallel? They'd be anti-parallel, and that would be called an anti-parallel beta-pleated sheet. Now, on the other hand, though, what if this guy here ran across, and then he just kind of looped off way over here and kind of sauntered his way around in the three-dimensional structure of the protein, and then came back right here and ran this way? Then this side would be running N to C left to right. The top would be running N to C left to right, and that would be a parallel beta sheet. And sometimes these just like hook back and forth like this with very small regions in between the parts that are actually linked in this fashion. So, and sometimes they do exactly what we did before and just go off somewhere else in the protein and then come back. So, and form the beta sheet. Both are common. Cool. So, question.
So, like in the helix, you said the hydrogen bonds are parallel to the helix. Could you say that these are perpendicular?
Yes, and that's the other important part here. So here, the beta sheet runs this way, but the hydrogen bonding is between sheets this way, and so it's perpendicular to the sheets themselves. Both those things are important. So for beta sheets, the hydrogen bonding is perpendicular to the sheets, the plane of the sheets, if you will. Whereas for the alpha helix, the hydrogen bonding is parallel to the axis of the helix. Cool. So again, big things to take away. Hydrogen bonding only involved in what part of the amino acid chain? Backbone. Only involving hydrogen bonding of the backbone for secondary structure. Question.
Can you show us how we can tell if that one's running end to C or student N?
So the way I've drawn it, you can't. But if you notice, take a look at it this way. So if you look at a typical amino acid structure, so I just draw a little segment here. You can totally tell which direction this chain is going, left to right or right to left. And the key is find your peptide linkage. Find your amide linkage. If you look here, there's my amide linkage. This side of the amide linkage comes from what part, the terminus of what part of an amino acid? The C terminus. This part, the carbonyl part, is from a carboxyl group. But the nitrogen's from what terminus? The N terminus. And so we see in this case, this end of this C hooked up to this N. And so there's an amino acid here, there's another one here. And if you look at one amino acid, this N is the beginning, that C is the end, I should really draw it there, of this one amino acid. And so you can see this one amino acid, the N's on the right, the C's on the left, that's where it's running. Same thing on this side. There would have been a nitrogen right here, and I was seeing for this one amino acid, N to C running to the left. So if you look at just this chain here, I didn't draw the top one in enough to make it really matter, but the bottom one in, is it running left to right or right to left?
From left to right. We've got C, right? Because it's where you've got your double bond to your oxygen. And
Awesome.
your current. So
There's one amino acid, if you will. So, and there's,
there's the next one, right? That's
awesome. And as long as you find one complete amino acid, you can totally go, oh, N to C.
so find the full one.
Yep. Cool. So tertiary structure. So we've gone past secondary structure. Tertiary structure really encompasses the entire folding, three-dimensional folding of a single peptide chain. A single peptide chain. But it's entire three-dimensional folding of it. And so now the R groups are involved. So for secondary structure, it is just the backbone. So, but now we're going to get into talking about the R groups are involved in the process as well for tertiary structure. So, and the interactions that can be involved, all of the above. Every interaction you've learned about, the intermolecular, which in this case would be considered intramolecular, all those that you learned about can be involved in the process here. So we can have electrostatic interactions. Notice if I have a lysine, what's the charge on a lysine at pH 7 in all likelihood? Positive. So, and somewhere else in the protein, I have an aspartic acid and aspartate. What charge is aspartate in all likelihood at pH 7? Negative. The lysine from one side could be attracted to the aspartate of the other side, and that plus-minus attraction is electrostatic interactions. And that could aid in the protein folding correctly and stuff like that. Cool. So electrostatic interactions can be involved in this. Hydrogen bonding. Notice there's a lot of amino acids like serine, threonine. Those are alcohols. Being alcohols, they have an OH, and that could involve hydrogen bonding with a serine in one part with some other polar hydrogen bond acceptor in another part of the protein. Can be involved in the folding. One of the big things involved in the folding, though, so was the hydrophobic interactions. What's hydrophobic interactions again? Only happens in water, in aqueous environment, and it's all the nonpolar residues doing what? Kind of coalescing together. So coalescing together. That's hydrophobic interactions, and that's a big part. So in fact, all those hydrophobic interactions, if you look at a lot of proteins, we'll talk about globular proteins specifically in a minute, they put all the nonpolar stuff in the middle and have all the polar stuff on the outside. Why would they do that? And act like they're thinking molecules, but why would they do that? Well, because all the outside polar ones can interact with the solvent around them, which is water. And that's great. Water loves them. They love water. And all the nonpolar ones get sequestered, we say, in the interior of that protein, avoiding water nearly entirely. It's a great situation, and the hydrophobic interactions play a large part in this. All those nonpolar ones coalesce in the middle. So in fact, if protein folding goes wrong, how would you think it might be easy to recognize that protein folding went wrong? If you have any nonpolar residues that didn't make it to the inside like they were supposed to. And there are proteins that recognize this, and they'll see that protein like, oh, that one's not folded right. They sense that nonpolar region that's on the outside. And they'll do one of two things. There are proteins that will pull that thing apart and let it refold. And other proteins that say, kill it. So things of that sort. So it's a danger, because oftentimes protein misfolding, that's where you often get your amyloid deposits in like Alzheimer's disease, Parkinson's disease, things of this sort. They don't know everything yet, but it often looks like it's due to protein misfolding, causing a lot of proteins to form these plaques that are just aggregates because nonpolar regions were exposed that shouldn't have been. Things of this sort. There's a little more to it than that, but that's the basis. Some of the research has been done. Cool. So we got electrostatic interactions, hydrogen bonding, so hydrophobic interactions, dipole-dipole forces. So we got another one in there. Covalent bonds can actually play a role. Covalent bonds can actually play a role. If you look at cysteine structure, so cysteine's R group has an SH, a sulfhydryl group. So it's a thiol. Some thiols do something funky. So if you have two cysteine residues in the same protein, if they can get close to each other, they may not be close to each other in the sequence, but if the whole protein folds over on itself, bringing them close to each other. So you might have another cysteine, from some other part of the amino acid chain down here or something like that. So, and cysteines can form what's called a disulfide linkage. So the disulfide linkage is technically considered an oxidation. You lose these two hydrogens and join the sulfurs together, a covalent bond. And this covalent bond can be part of the protein folding process. So in fact, we often, to get proteins to unfold, we'll add a reducing agent. And the reducing agent would turn this guy back into this, untethering these two parts of the protein, of the peptide. Cool. So, but again, when they come together during protein folding and form that covalent bond, that's your disulfide bridge. And so we see that pretty much every kind of interaction you can think of is involved in tertiary structure. Not just intermolecular forces, but even covalent bonds can be involved. Now, not all proteins will have quaternary structure. A lot do, but not all do. So first part, quaternary structure, everything that is involved in tertiary structure is involved in quaternary structure. All the interactions we just talked about, they're all part of quaternary structure as well. Here's the only difference. For quaternary structure, this is the interaction between different peptide chains. So a lot of proteins actually are made of multiple peptide chains. And how those peptide chains interact with each other is the quaternary structure. And those two different peptide chains could have disulfide bridges between them, hydrogen bonding, electrostatic interactions, all that stuff. Everything that played a role in tertiary structure also plays a role in quaternary structure. Somebody named for me a protein where the quaternary structure was really important in this chapter. Hemoglobin. So how many peptides are in hemoglobin? 4. You got 4 peptides. 2 alphas, 2 betas, right? So two are identical, but all 4 come together and interact all together with quaternary structure. Now notice, not all proteins do this. Some proteins, they act alone. They're loners. I don't work with anybody. I'm just going solo on this one. Well, if they go solo, they don't have quaternary structure. So, but if any proteins form a larger protein structure with other peptides, that is quaternary structure. Cool? Cool. This quaternary structure, we'll see with hemoglobin a little bit, that often results in things like cooperativity. In hemoglobin's case, that'll be positive cooperativity, but we won't see it, but there are examples of negative cooperativity as well. So these are all your levels of structure in a protein. So real quick review. Primary structure is pretty much just only what? amino acid sequence, but all the info for what is involved is provided there for folding. Secondary structure, what's involved? Hydrogen bonding with only the backbone. Sweet. Tertiary structure, ultimate three-dimensional shape of a single peptide, and everything's involved. And quaternary structure, just interactions between multiple peptide chains, and again, everything's involved. Awesome. Keon.
Could your backbones of one peptide have attraction to the backbone of another peptide?
Could the backbone of one peptide interact with the backbone of another peptide? Yes. So, and the most common example is when beta sheets form from separate peptides. So it's a little less common, but that can happen. So, but yeah.
Is that part of quaternary?
So it would still be considered part of quaternary. So it kind of might also be considered part of secondary as well if it formed like a beta sheet. But technically, it will fit under the umbrella of quaternary if they're from separate peptide chains. Kevin.
This is a little off what we're talking about, but in the lecture notes, there's a study problem about the tertiary structures, and they give two amino acids like phenylalanine and isoleucine.
Oh, and ask how they relate or what interaction they would have with each other?
Yeah.
Sweet. Cool, you totally know. And actually, I think you got all the tools you'd need to know. So let's say I give you any two nonpolars. Let's just say I give you valine and leucine. Well, they're nonpolar. What's the most important interaction of nonpolar residues for tertiary structure for protein folding? Well, don't go there. That's an intermolecular force, which is, it definitely is part of it. But again, this is something that only happens in an aqueous environment that causes all those residues to get in the middle of a globular protein, let's say. What is that interaction? Hydrophobic interaction. So if you get two nonpolar residues, the most important thing for causing them to interact with each other is the hydrophobic interactions. What if I gave you a lysine and an aspartic acid? Lysine and aspartate. Well, lysine's what kind of amino acid? Basic. Aspartate's what kind? Acidic. Lysine at pH 7 has what charge? Positive. And aspartate has what charge at pH 7? Negative. Guess what kind of interactions between those two? Electrostatic interactions, plus minus interactions. So let's say I gave you serine. Actually, let's just say I gave you any two in the polar column. Any two of those: serine, threonine, tyrosine, asparagine, glutamine, cysteine. All of those can be involved in hydrogen bonding. And so if I give you any two of those, they're going to interact via hydrogen bonding. So on and so forth. And so that's pretty much why she wanted you to memorize each category. That way, if you look at a couple of interactions, now you know what the major tertiary structural element that's keeping them together is.
Well, what if it's like polar and acidic?
Well, let's look at polar and acidic. Is polar and acidic going to be a plus-minus thing? No. It's not going to be a plus-minus thing. So then what kind would it be left at that point? Is it going to be hydrophobic? Definitely not. Definitely not hydrophobic. It's still hydrogen bonding at that point as well. So they both have either donors or acceptors or both for hydrogen bonds. So it'd definitely be hydrogen bonding in that case.